all love reading storybooks. Every story is framed with sentences, and sentences are framed using words. And what are words made up of? They are made up of letters. If you notice, then everything around us is made up of one or the other smaller entity. These small entities are called basic units. Did you know that even our body is made up of basic units? Yes, it is. Although it appears to be one single body, it's actually made up of millions of basic units coming together. What are these units called? They are known as cells. Cells come in different numbers to form a body, rather, any living organism's body. Can you guess the minimum number of cells required to form an organism's body? Do you know the answer? There are organisms which are made up of only a single cell. They are called the unicellular organisms. Bacteria are one of the best examples of this category. What are organisms like us called then? We belong to the category of multicellular organisms. Those having several cells to make up the body are called multicellular organisms. Now the next important question is, if there are so many cells in our body, then are all of them exactly the same? Of course not. There are many number of cells in our body, and so are their types. Look at these cells, for example. These look so different, but astonishingly, all three are a part of our body. This one is an ovum, this is a neuron, and the last one is a red blood cell. Their structures, shapes, sizes are totally different from each other, and so are their functions. This is how various types of cells come together in different numbers to form our body. We have this knowledge today, but ever wondered how all this was actually discovered? Let us go back in time and have a look at the history of cellular studies. If I ask you what forms the basic unit of every organism's body, I'm sure you'll agree that it's cells. This seems obvious now. But how was this discovered? How were cells discovered? Who worked on cells to understand them so well? Let's answer these questions. For that, let's get back in time and begin with understanding the milestones in the history of cell studies. The time dates back to around 1665, when a legendary English scientist named Robert Hooke came up with an interesting concept. The era was ruled by newly invented instruments which can be related to the present-day microscopes. Robert Hooke, with a self-designed microscope, observed a thin slice of a tree cork. What did he observe? He found tiny structures clustered together. An image something like this. To him, these structures appeared empty from within and had prominent borders. Do they resemble any structure? At first glance, they resemble the honeycomb structure. But for Robert Hooke, these structures appeared like the small rooms in a monastery where the monks lived. With this in mind, he named these empty structures cells. Why cells? The literal meaning of the term cells was the empty rooms where monks or nuns lived. That is why he named these as cells. So he was the one to coin the term cells. Although his discovery was brilliant, do you think this much information is sufficient to study about life? The cells seen by him were like empty boxes. So what next if there is nothing inside the cell? There's a lot more. Let's move a bit forward in this timeline. Fascinated by glasses and lenses, a Dutch businessman made his own microscope. He was none other than Anton van Leeuwenhoek. Most commonly referred to as the father of microbiology, he came up with an outstanding model of a self-designed microscope which is believed to be better than many microscopes available today. The height of his fascination made him check even the dental scrapings under the microscope. Although it sounds weird, that is how he became the first person to observe live cells under a microscope. What do you think he must have observed in the dental scrapings? He observed numerous tiny bacteria, but he did not name them so. Instead, he addressed them as animalcules because for him they resembled animals. This is how progress was achieved in the history of cell studies. Robert Hooke observed the cells first and coined the term. Similarly, Anton van Leeuwenhoek observed live cells for the first time. However, this was about the history, how cells were discovered and how they appeared under the efficiently designed microscopes. Cells form the basic units of life. We know this. And by now, we even know how the discovery of cells, as seen in the tree cork, was a breakthrough in biological studies. Now it's time to have a look at how the cells actually look under a microscope. For this, we select onion peel as our specimen. But before we begin with the procedure, let's first collect the necessary requirements. We will need a microscope, a slide, a cover slip, iodine solution, water, and forceps. Anything else we're left with? Well, the most important of all is the specimen, that is an onion peel. Now that we're all set with the equipments needed, let's begin with the amazing experiment of observing the onion cells under the microscope. Firstly, we take the onion peel and with the help of the forceps, pull away the thin lining from the inner surface. Before placing this on the slide, we first add a drop of water on, then place the onion peel on it. We are not supposed to touch the peel and even the slide with our fingers. Next, we add 2 drops of dilute iodine solution. Hold on, do you know why is iodine solution added to this specimen? We do this so that we can clearly see all the parts of the cell. Else it's difficult to study them as they are transparent. It would even be difficult to differentiate between the various parts. Now we place a cover slip in this manner and make sure that no air bubbles are formed on the slide. With this, our slide is ready. This entire process of setting up the microscope along with the slide is termed mounting. It's also important to see that there is enough light falling on the mirror of the microscope. Shall we observe it now? Here we go. Wow, doesn't it look amazing? Now before studying these, tell me one thing. Do plants and animal cells look the same? Do they have the same organelles? Or are the 2 types different in some ways? Well, plant and animal cells are slightly different. When cells from a human cheek are scraped with a sterile cotton bud and mounted, similar but not exactly same cells are seen under the microscope. Roughly, we have these thick, distinct borders that differentiate each cell from the other one. Then we have these thin, tiny dark spots scattered all over. Also, we can spot this prominent circular structure here, which is very darkly stained. Any ideas what these things are? Well, these are the cell constituents. For our convenience, let's select just one. Say the onion peel cells to understand the structure and the contents of the cell. Roughly, we've seen these thick, distinct borders that differentiate each cell from the other one. Then we have these tiny dark spots scattered all over. Also, we can spot this prominent circular structure here, which is very darkly stained. Any idea what these things are? Well, these are the cell constituents. Let's get introduced to each. This outer margin seen here is called the cell wall. If we zoom in a bit, we notice that there is another layer in the inner side of this cell wall. This is called the cell membrane. Yes, the outer wall is the cell wall and the inner layer is called the cell membrane. The cell membrane is also referred to as the plasma membrane. It is porous in nature. So what does porous mean? It simply means that any material can move in and out of the plasma membrane. But isn't that dangerous? Of course it is. But the simple solution lies in the nature of the plasma membrane. That is because it is selectively permeable. So what does selectively permeable mean here? Just like a guard standing at the office door who allows only members with the identity cards to enter and exit, the plasma membrane also allows only specific materials to move across. Do you think all cells have cell walls? Well, the answer is a no. That's because animal cells do not have cell walls. An animal cell only has the plasma membrane and not the cell wall. The cell walls act as protectors for plant cells. It's composed of a non-living rigid structure that is cellulose. It not only acts as a protector, but also gives a proper shape to the plant cells. It also helps in cell-to-cell interactions and provides a barrier to undesirable macromolecules. It protects, it gives shape to the cells, and it helps in cell-to-cell interactions. Clearly, the cell wall is a very important part of the plant cell structure. Now let's get into this lighter region here. This lighter region is the cytoplasm. It's a jelly-like substance filling up the cell space. And these tiny parts in the cytoplasm are the organelles. And what exactly are they? Just like the human body has different organs, the cell has various organelles. And just like the organs, each organelle performs a different function. If we are given this image and asked what exactly this is, I'm sure your answer will be a cell. Now to study this, we need to study every component present within. And not only within, we even need to study the covering, that is, this part in detail. Now, can you tell me what exactly is this named as? That's right, it's called the cell membrane or the plasma membrane. In our previous video, we have learned that the cell membrane is selectively permeable in nature. That is, it only allows specific materials to enter and leave the cell. Any guesses what is the plasma membrane made up of? The plasma membrane is made up of lipids and proteins and also has some carbohydrate chains. There are two things that freely move in and out of the cell. Those include gases and water. Do you think these molecules need energy for traveling across the membrane? Let's take a simple example to understand what exactly happens in this case. We know what happens when a drop of ink is added to a glass of water. The single dark-colored ink drop spreads when added to water and the intense blue color fades slightly. If we see at the molecular level, then the ink molecules, which are compactly packed in the single drop, get a chance of dispersing in a larger area when added to the water. This is nothing but the process of diffusion. And we know that this dispersal process does not require energy. The molecules just try to move freely in the space available, and as a result, diffusion occurs. The same story is seen when gas or water molecules travel across the membrane. Take carbon dioxide for example. When cells undergo cellular respiration, a lot of carbon dioxide accumulates in the cell. On the other hand, there is very little carbon dioxide present outside the cells. Hence, the carbon dioxide molecules move outside the cells. Now, there is very little oxygen left after respiration in the cells. So the oxygen from outside the cells moves in through the plasma membrane to make up for the lost oxygen. That means carbon dioxide moves out and oxygen moves in. Both the gases move from their area of higher concentration to an area of lower concentration. And this diffusion process does not require energy. Diffusion is one of the processes by which transportation of several substances is carried out across the cell membrane. Now let's get to know one more interesting concept named osmosis. Have we ever come across this term osmosis? Well, quite often. But what exactly do we mean by this? Osmosis is defined as the movement of water from higher water concentration to lower water concentration through a semi-permeable membrane. So let's say we have this container which is partitioned by this semi-permeable membrane. Now we add sugar solutions at both these sides. However, the concentration of each is different. Here we add concentrated sugar solution, while here dilute sugar solution. That means the concentration of water in this solution is obviously more compared to this. So after some time, we see water molecules moving from this side of the partition to this side. And how long will this movement take place? It will occur till the concentration of water is balanced, that is, equalized on both the sides. This is nothing but osmosis. We all have seen raisins swelling up when kept in water for some time. Any idea how this magic works? Well, it's simply the process of osmosis which results in the swelling of these raisins. You heard me right. It's osmosis process due to which the water from the surrounding medium enters the cells of the raisins, helping them to swell. Can we define osmosis once? It's simply defined as the process in which water moves from higher water concentration to lower water concentration through a semi-permeable membrane. An interesting thing to know about osmosis is that the movement of water is affected by the amount of a substance dissolved in it. And what does this mean exactly? Let's conduct an experiment to understand it better. Let's take 3 plates or containers with salt solutions of different concentration in each. Let's add a few red blood cells, that is RBCs, in each. After a while, we notice that the first set has cells which are shrinked, the second set has cells that are just the same, while the third set has cells which are swollen or bulged. Any idea why this happened? Let's understand this. In the first case, the water content in the surrounding solution was less as compared to the cells placed in it. In other words, the salt content of the solution is more, and therefore this solution is a concentrated one. That is the reason why the water molecules moved from inside the cells to the outside solution, as the concentration of water was higher within the cells. Hence the cells started shrinking. This kind of solution is called as hypertonic solution. That is the solution with higher salt concentration. Now let's have a look at the second case. Here, the level of salt and water seems to be the same on either side of the cell membranes. That is, both within and outside the cell. Therefore, there was a balance in the movement of water. What do we understand by this? It means that the movement of water was the same in either directions. As a result, the size of the cell did not change. It remained the same. Do you know what such a solution is called? It's called as isotonic solution. The two solutions are isotonic when the concentration of water is the same on either sides of the semi-permeable membrane. Now let's focus on this third set. The cells in this case are surrounded by a dilute solution, that is by higher water concentration. The salt dissolved is less and the water content is higher. Therefore, water molecules move inside the cells and hence the cells get swollen. Such a solution is called a hypotonic solution. In other words, solution with a higher water concentration compared to the other. This is how various solutions and their different concentrations help cells to sustain pressure and to survive. Movement of molecules across the plasma membrane is also influenced by the simple process of osmosis. Any idea how a factory works? Well, a factory which manufactures many things carries out several functions like assembling, modifying, packaging, and then transporting the manufactured goods. A similar process takes place in a cell. This is the organelle of our focus. The endoplasmic reticulum works as a factory that manufactures certain important substances. What functions do ribosomes serve? Well, ribosomes are the sites for protein synthesis. That means the place where proteins are actually made. Organelle, that is Golgi apparatus. This unit is called as the modification and packaging unit of the cell. The apparatus is named after Camillo Golgi, who was the first to describe the Golgi apparatus. He found a revolutionary method of staining individual nerve cells and studied the structures of the cells.